During the mid-Miocene, about 15 million years ago, a region of central and southeast Italy around Gargano and Scontrone was cut off from the mainland by rising sea levels. One of the strangest residents of the island was Hoplitomeryx, an early type of ruminant that resembled a deer or pronghorn. Multiple species have been identified, representing 4 different size classes ranging from huge down to tiny insular dwarfs. Each of these size classes was specialized for slightly different ecological niches, eating different types of vegetation to avoid directly competing with each other for the limited amount of food on the island. Chevrotain family was widespread and successful from the Oligocene through the Miocene, but has remained almost unchanged over that time and remains as an example of an archaic ruminant type. They have 4-chambered stomachs to ferment tough plant foods, but the third chamber is poorly developed. Unlike other artiodactyls, they lack a carotid reedy, and so cannot heat exchange cool blood entering their brains, a thermoregulatory innovation that allows other artiodactyls to exploit hot arid habitats. Though most species feed exclusively on plant material, the water chevrotain occasionally takes insects and crabs or scavenges meat and fish. The water chevrotain makes several different noises, which include a scream when injured, wounded, and an alarm bark. When females fight, they make a high-pitched chattering noise, and when pursuing a female, a male makes a noise through a closed mouth. The greater mouse deer is solitary and nocturnal. It uses small trails through thick brush in the forest. When the male is ready to mate, he rubs a large gland on his lower jaw against the female to determine whether she is ready to mate. If she is not ready, she responds by walking away. The male is very territorial, marking his territory with feces, urine, and secretions from the intermandibular gland under the chin. When angry, the male beats the ground with his hooves at a rate of 4 times per second. They are rather trusting but delicate animals. They feed on fallen fruits, aquatic plants, leaves, shrubs and grasses. Protocerus was 1 meter long and had a body shape similar to that of a deer. Like some other protoceratids, it had 3 pairs of blunt horns on its skull. These horns were likely covered with skin in life, resembling the ossicones of a giraffe. It exhibited sexual dimorphism. Females had only 1 pair of horns located at the back of the skull, which were shorter than the corresponding pair in males. Males likely used these horns for display purposes, either to attract females or intimidate rivals. Due to the orientation of the horns, males probably displayed them sideways rather than frontally. Protocerus was one of the earliest and most primitive protoceratids, still retaining upper incisors and 4 functional toes. It lived in the deserts of the late Oligocene. The skull decoration of Syndioceras was quite unlike that of a deer. It had 2 pairs of horns, a V-shaped pair on the snout, fused at the base, and a second pair positioned between the eyes and ears, curving inward in a semicircular shape. Like the ossicones of a giraffe, these protrusions were covered with skin and were likely used for display and fighting. In addition to its horns, it had tusk-like canine teeth, which it may have used to root through soil and undergrowth for food, similar to a modern musk deer. The shape of its skull also suggests it may have had an inflated muzzle, resembling that of a modern saiga. This 1.6-meter animal closely resembled a deer, with 2 hooved toes and a weight exceeding 60 kilograms. Like early horses such as Merychippus, it had 2 vestigial outer toes on each foot that did not touch the ground. Synthetoceras was one of the largest protoceratids, standing about 1 meter tall at the shoulder. Its 2 nose horns were partially fused into a single long structure with a forked tip, which may have been used for sparring in a similar manner to the antlers of modern deer. With a length of 2 meters and a mass of 200 kilograms, Synthetoceras was the largest member of its family. It was also the last, and had what is considered to be the Protoceratid's strangest set of horns. The two horns above its eyes looked fairly normal and similar to those of many modern horned mammals, but on its snout, it had a bizarre, long horn with a forked tip that gave it a Y shape. Only males had this strange horn, and they probably used it in territorial fights. Kyptoceras is the last known member of the family. The Protoceratids were believed to have been driven to extinction by more advanced grazing herbivores, but in Florida, where there were still relatively large tracts of forest, the Protoceratids were able to survive. The genus name comes from its bent horns, in which the two horns above the eyes curved over its head, and the two nasal horns pointed forward. Hexameryx had 6 horns, an unusual trait among ungulates. These horns were arranged in 3 pairs, 1 pair on the snout and 2 pairs above the eyes. This distinctive feature sets Hexameryx apart from other members of the Antilocapridae family. In most respects, this family resembles other ruminants. They have a complex, 4-chambered stomach for digesting tough plant matter, cloven hooves, and small, forked horns. Their horns resemble those of the bovids, in that they have a true horny sheath, but, uniquely, they are shed outside the breeding season and subsequently regrown. Their lateral toes are even further diminished than in bovids, with the digits themselves being entirely lost, and only the cannon bones remaining. Ilangoceros is known for its unique horn structure. Unlike modern pronghorns, it had long, spiraled horns that curved backward. This distinct horn shape is one of its most recognizable features. It lived during the Miocene epoch, approximately 10 to 15 million years ago. This was a period characterized by significant changes in climate and vegetation, which influenced the evolution of many mammal species. Ilangoceros inhabited the open plains and grasslands of North America. These environments provided ample grazing opportunities and influenced the development of its physical adaptations. Its teeth were adapted for grazing on grasses and other vegetation, which was abundant in its Miocene habitat. As part of the Antilocapridae family, it is an important genus for understanding the evolutionary history of pronghorns. Its unique horn structure and other anatomical features provide insights into the diversification of this group. Asymmetry is commonly seen in the headgear of modern even-toed ungulates, with natural genetic variation, developmental stress, and injuries during life sometimes causing very wonky-looking horns or antlers. No living species have asymmetry as a standard trait, however, but some fossil ungulates did. Rhinoceros was smaller than modern pronghorns, around 70 centimeters tall at the shoulder, and males had long antler-like horns with 3 tines. Bizarrely, one of these horns was always at least twice the size of the other, with left-horned and right-horned individuals seeming to occur in equal numbers. It's not clear how this asymmetry affected combat between males. Could they only properly lock horns with opposite-sided rivals, or did this uneven arrangement actually prevent physical fights and restrict them more to just visual displays? 5 species of Tetrameryx are known, but one species, Tetrameryx schulleri, survived until about 12,000 years ago, and was present when Paleo-Indians reached North America. Although approximately the same size as the living pronghorn, the limb bones are somewhat more robust, suggesting that its running speed was slower than living pronghorn. Tetrameryx is distinguished by having 4 horns, arranged in 2 pairs. This contrasts with the single pair of horns seen in the modern pronghorn. The 2 pairs of horns were positioned with 1 pair on the front of the skull and another behind them. Pronghorn possess large eyes with a 320 degrees field of vision and a unique dental formula. Unlike deer, pronghorns have a gallbladder. Males have horn sheaths that can grow up to 43 centimeters, while females have smaller, sometimes barely visible horns. Pronghorns are the fastest land mammals in the Western Hemisphere, capable of sustaining high speeds for long distances, capable of reaching 88 km per hour for short distances and sustaining lower speeds over longer distances. They likely evolved this speed to evade now extinct predators. Despite their speed, they cannot jump well and often crawl under fences, prompting modifications to fences to aid their movement. Pronghorns signal danger by flaring the white hair on their rumps, releasing a scent reminiscent of buttered popcorn to alert others. They form mixed-sex herds in winter, with social structures changing in spring. Males either defend territories or harems, and females have various mating strategies. They breed in mid-September, with females giving birth after a 7 to 8 months gestation. Newborns spend their early days hiding in vegetation, with mothers providing care and protection. Pronghorns reach sexual maturity at 15 to 16 months, with males rarely breeding before 3 years old. Their lifespan typically reaches up to 10 years, sometimes extending to 15 years. Cranioceras is an extinct genus of endemic artiodactyl from the grassy plains of what is North America, during the Miocene epoch. This unusual deer-like creature had 2 straight horns above the eyes and a single curved horn on the back of its head. Little is known about these animals, however, since they very rarely are studied in the paleontological record. They may have been dense woodland and open bushlands grasslands dwellers that mainly browsed on non-grass vegetation. Their gradual decline seems to have been the result of disappearances of suitable habitats as a result of increasing aridity and cooling climates. Ampelomyrix, a Palaeomerycid ungulate from the early Miocene of France. About the size of a deer, around 1 meter tall at the shoulder, it was a distant relative of modern Giraffes. Males sported 3 distinctive ossicone-like horns, 2 over their eyes and a third forked one at the back of the skull, and protruding tusks like some modern deer, which probably served a similar purpose in fights against each other. In primitive members of the group, this appendage was a posterior expansion of the occipital bone lying close to the powerful muscles supporting the skull in a normal position, thus suggesting that this appendage was actually used for fighting between males during the breeding season. The reduced shapes of the flat and laterally oriented appendages of later species suggests that these were not used in active fighting, instead forming a function of passive display. In some species this horn structure formed a single central horn shape, while in others it forked out to each side. They also often had long saber-like canine teeth similar to modern water deer and musk deer, which were probably used for fighting while their elaborate headgear was purely for visual display. Xenocerix is an extinct genus of ruminant known from the Miocene of Europe. The type species, Xenocerix amygdalae, was recovered from central Spain and bears a unique T-shaped protrusion from the top of the head. The specific epithet amygdalae is in reference to the character Padme Amidala from the Star Wars films due to the striking resemblance that the occipital appendage of Xenocerix bears to one of the hairstyles that the aforementioned character shows in the Phantom Menace feature film. Prolibytherium living in North Africa during the early to mid-Miocene, about 17 and 16 million years ago. Its exact evolutionary relationships are uncertain, but it was probably part of a group called Climacoceratidae, deer-like giraffoids which often had thorny branching ossicones that resembled antlers. It stood around 1.2 meters tall at the shoulder, and exhibited dramatic sexual dimorphism. Females had slender forked horn-like ossicones, while those of the males flared out into large wide flat shapes that resembled butterfly wings. Heavy reinforcement in the bones of the back of the male's skulls helped to support all the extra weight of those huge ossicones, and if they actually used the structures to fight with each other, then this may have also provided some protection or shock absorption. Discocaryx had a thick-boned cranium which had disc-shaped headgear located in the middle of the head, cervical vertebrae with thickened centra, and the most complicated head-neck joints in any mammal known at the time of its discovery in 2022. These adaptations were for head-butting behavior between males, comparable to the behaviors of rams and musk oxen as well as the neck-blowing in modern male giraffes. The neck adaptations of Discocaryx help scientists to better understand the triggers for the evolution of the necks of giraffoids. Compared to extant head-butting animals such as rams and musk oxen, it had the most optimized head-butting adaptations of all, with a skull that protected the brain more efficiently than other head-butting mammals. Tooth enamel isotopes indicate that the species was an open-land grazer which drank from multiple sources of water, and that their habitats included areas that other contemporary mammals were not adapted to make use of like Discocaryx could. Climacoceratidae is part of the Climacoceratidae family, which is closely related to the Giraffidae. This relationship highlights the evolutionary experimentation with horn and body shapes in this lineage. As a herbivore, it would have fed on a variety of plant materials available in its environment. The structure of its teeth suggests it was adapted to browsing on leaves rather than grazing on grass. It is likely that it exhibited sexual dimorphism, with males having more prominent and elaborate horns compared to females. This trait is common among horned ungulates and is typically related to mating displays and competition. While Climacoceratidae shares some characteristics with modern giraffes and deer, its unique horn structure sets it apart. Studying these differences helps scientists understand the evolutionary pathways that led to the diversity of horned mammals we see today, in that their antler-like ossicones are derived from different bones. Palaeomerycidae is considered one of the early members of the Giraffidae family. Its fossil record extends back to the Miocene epoch, indicating its presence during a period of significant mammalian diversification. Like modern giraffes, it likely had ossicones that were present in both males and females. These ossicones were probably covered with skin and hair, similar to those seen in okapis. It inhabited diverse habitats during the Miocene, ranging from open woodlands to savanna-like environments. This suggests it had adaptations suited for browsing on leaves and vegetation. Phylogenetic studies place Palaeomerycidae in a close relationship with both cervids and bovids. This indicates shared ancestry in evolutionary pathways, possibly reflecting adaptations to similar ecological niches and dietary preferences. Giraffidae is distinguished from other giraffids by the 4 ossicones on its head, one pair in front of the eyes on the anterior aspect of the frontal bone and the other behind the eyes in the frontoparietal region overhanging the temporal fossae. A comprehensive taxonomic analysis of fossil ruminants from 3 sites within the Chinji Formation has been conducted. These sites have yielded a significant quantity of fossil fauna dating to the Middle Miocene epoch. Among the newly collected ruminant samples are representatives from 3 different families, Tragulidae, Giraffidae and Bovidae. Notably, the skull of Giraffidae has been documented for the first time at the Dak Bon Emir Khatun locality. A medium-sized member of the Giraffidae family, it is renowned for its characteristic 2 pairs of horn cores. The lithostratigraphic and paleo-environmental data from the Chinji Formation suggest that the Miocene climate in Pakistan was likely monsoonal and humid, featuring extensive forests and grasslands. Giraffes are renowned for their distinctive long necks, a characteristic absent in their closest living relative, the okapi. An intriguing intermediate fossil, Samotherium from Samos Island, Greece, lived approximately 7 million years ago. This species has long fascinated researchers as a transitional form showing traits suggestive of giraffe neck elongation. Comparisons of the 1-meter-long neck bones with those of modern giraffes and okapis revealed intriguing insights. The team noted that Samotherium's 2nd and 3rd vertebrae exhibited mixed features resembling both giraffes and okapis, while the 5th to 7th vertebrae showed transitional characteristics. Notably, the sixth vertebra displayed a surface ridge akin to an okapi, distinguishing it from the giraffe. Detailed anatomical analyses concluded that while it is not a direct ancestor of modern giraffes, it is a close evolutionary relative. The okapi stands about 1.7 meters tall at the shoulder, with a body length averaging 2.5 meters and weighing between 200 to 350 kilograms. Its striking appearance includes a long neck, large flexible ears, and a coat ranging from chocolate to reddish-brown, adorned with zebra-like stripes on the legs and ankles, serving as camouflage in dense vegetation. Both sexes have interdigital glands on all 4 feet, with males additionally possessing short, hair-covered ossicones less than 15 centimeters long, akin to those of giraffes but absent in females. Adapted to its tropical habitat, the okapi boasts numerous sensory and physiological adaptations. It has a high concentration of rod cells for night vision, large auditory bulli for acute hearing, and a well-developed olfactory system. Its dental formula and teeth morphology are specialized for efficiently cutting tender foliage, aided by a large cecum and colon facilitating microbial digestion and quick food passage. Okapi exhibits sexual dimorphism and shares more external similarities with bovids and cervids. Ecologically, okapis are primarily solitary and diurnal, preferring dense forests at elevations of 500 to 1500 meters. Their diet consists of a variety of vegetation, selectively browsed using their lengthy tongues. Despite being endemic to a restricted range, okapi populations face significant threats, leading to their classification as endangered. Habitat loss due to logging and human settlement, hunting for bush meat and skins, and illegal mining pose major conservation challenges. Shansatherium possessed a unique appearance characterized by relatively short legs and a long neck, similar to modern giraffes but with distinct differences in proportions compared to other early giraffids. It was moderately sized for a giraffid, indicating it likely exhibited a lifestyle somewhat different from both modern giraffes and other extinct relatives. It is believed to have inhabited forested environments, contrasting with later giraffids that adapted to more open habitats. During its existence, the regions where Shansatherium lived in Asia likely had a mix of forested areas and open landscapes, contributing to its unique evolutionary trajectory within the giraffid family. Civatherium, originating in the late Miocene around 7 million years ago in Africa, persisted until the late early Pleistocene. It resembled a larger, more robust version of the modern okapi, standing about 2 meters tall at the shoulder and reaching a total height of 3 meters, with a weight up to 1,000 kilograms, making Civatherium among the largest known ruminants, comparable in size to modern giraffes and large bovines. These estimates may be conservative, not accounting for the substantial horns found in males. It had a distinctive set of wide, antler-like ossicones on its head, with a second pair above its eyes. Its robust shoulders supported the neck muscles necessary to lift its heavy skull. Initially mistaken as an evolutionary link between modern ruminants and the now disregarded pachyderms, it was notable for its graviportal, or robust, morphology unlike any other known species at the time. Civatherium was a mixed feeder capable of both grazing and browsing. Brahmatherium was built very similarly to Civatherium. Alive, it would have resembled a heavily built okapi or a moose, and had a crown-like set of 4, radiating ossicones. Fossils, and examination of teeth in particular, suggested the living animals dwelled woodlands and wetlands. Like other giraffids, Brahmatherium had long legs and a long neck adapted for reaching vegetation high off the ground. Most probably, these horns had a demonstration function. However, it is possible that they were used in males' duels, with their solid bases and rear pair directed sideways. Brahmatherium, like many other large mammals of the Miocene epoch, became extinct as environmental conditions and habitats changed over time. Decenitherium is part of the evolutionary lineage that eventually led to modern giraffes. Its anatomy and adaptations offer clues about the evolutionary transitions within giraffids, including changes in body size, feeding strategies, and habitat preferences over millions of years. It coexisted with a diverse range of other mammalian species during the Miocene, including early forms of elephants, rhinos and other ungulates. Studying its fossil record helps researchers understand the broader dynamics of mammalian communities in ancient Europe. Significant fossil discoveries of Decenitherium have been made in France and Spain, where well-preserved remains have been recovered from various geological formations. These discoveries continue to contribute to our understanding of giraffid evolution and paleoecology in Europe. Balinia was a genus of giraffid known from the late Miocene of Europe and Africa. It was remarkably similar to the modern giraffe of the genus Giraffa, with long limbs and a long neck, and may have been ancestral to it. Balinia is different from its modern relative in having a sloping skull and longer, horn-like ossicones. The elongation of the neck in giraffes is attributed to natural selection pressures for accessing higher branches and foliage, a strategy that likely evolved to avoid competition with other herbivores. Overall, the evolutionary journey of giraffes underscores their adaptation to specific ecological niches and highlights their unique position among modern-day mammals. The Rothschild's giraffe is easily distinguishable from other subspecies. The most obvious sign is in the coloring of the coat or pelt. Whereas the reticulated giraffe has very clearly defined dark patches with bright whitish channels between them, Rothschild's giraffe more closely resembles the Maasai giraffe. However, when compared to the Maasai giraffe, the Rothschild's ecotype is paler, the orange-brown patches are less jagged and sharp in shape, and the connective channel is of a creamier hue compared to that seen on the reticulated giraffe. In addition, Rothschild's giraffe displays no markings on the lower leg, giving it the impression of wearing white stockings. Rothschild's giraffes mate at any time of the year and have a gestation period of 14 to 16 months, typically giving birth to a single calf. They live in small herds, with males and females living separately, only mixing for mating. The Rothschild's giraffes are tolerant of other animals around them as long as they don't feel threatened. For the most part, they are very friendly, but the males are known to engage in fights for mating. Since this species can mate all year long, those battles seem to be frequent. The distinctive patches or spots on giraffes' coats serve several purposes and have fascinated scientists studying their evolution. The patches help giraffes blend into their surroundings, providing effective camouflage in the dappled light of their savanna habitat. It's also theorized that the patches may play a role in regulating body temperature. Studies suggest that the different pigmentation in the patches affects how heat is absorbed and reflected, potentially helping giraffes manage their body heat in the hot African sun. The evolution of these patches likely involves both natural and sexual selection pressures. Natural selection favored patterns that enhanced survival through camouflage and thermoregulation, while sexual selection may have influenced the development of more elaborate patterns as a display of fitness. The patch's size, shape, and distribution vary among giraffe subspecies, reflecting genetic diversity and adaptation to local environments. The Maasai giraffe can be found in central and southern Kenya and in Tanzania. It has distinctive jagged, irregular leaf-like blotches that extend from the hooves to its head. Reticulated giraffe is differentiated from other types of giraffes by its coat, which consists of large, polygonal, block-like spots, which extend onto the lower legs, tail and face. These prominent liver-red spots also show much less white between them, when compared to other giraffe species. They are diel, meaning they are active both day and night. They are most active during dawn, dusk and midnight due to their warmer environment, a behavior that can also be described as crepuscular. Their sleep patterns are short, typically no more than a few hours at a time, usually standing up. Reticulated giraffes are herbivores, feeding on leaves, shoots and shrubs, and spend about 13 hours a day foraging. As ruminants, they practice foregut fermentation, which aids in digesting their high-fiber diet. In giraffes, the left recurrent laryngeal nerve is over 2 meters long, making it the longest in any living animal. This nerve begins in the brainstem, travels down the neck with the vagus nerve, and then branches off to loop back up to the larynx, giving some nerve cells a length of nearly 5 meters in the largest giraffes. Despite its long neck and large skull, the giraffe's brain is typical for an ungulate and is kept cool by evaporative heat loss in the nasal passages. The giraffe circulatory system has special adaptations, such as a thick-walled heart and a rete mirabile, to manage the high blood pressure needed to pump blood to its brain and to prevent fainting when it raises or lowers its head.